Welcome to Longevity.Technology Unlocked, where we explore the science and tools that help you live healthier for longer. I'm Dr. Nina Patrick.
And I'm Phil Newman. Now, Nina, when people hear about stem cells, they could assume it's all one thing. So today we're diving into the topic that sits right at the heart of next generation medicine, and that's stem cells, but different types, because specifically there are differences between autologous and allogeneic approaches.
Yes, and it really matters where those cells come from. So autologous means using your own cells. Your body is both the patient and the donor, while allogenic means using cells from someone else, a separate donor.
Yeah, so allogenic's been getting a lot of attention lately because it lends itself obviously to scale. You know, if you can find the right donor cells, you can get a kind of off-the-shelf product, one donor supplying treatments for many patients. So that's why we're seeing such an explosion in allogeneic approaches with donor sources from like cord blood to non-viable ectopic pregnancies, even clean living 20-somethings.
Yeah, autologous therapies, on the other hand, are much more personal, and that means they're more complex. You have to take the cells from each individual patient, process them, return them, which is logistically harder, it's more time consuming, and it can be more expensive. But the flip side is huge potential benefits in safety, immune compatibility, and truly personalized regenerative medicine.
Yeah, and that's exactly why we're so intrigued by our two guests today. We're talking to two founders who are really pioneering sophisticated autologous technologies that could make personalized cell therapies really drive health span and longevity.
That's right, Phil. These founders have started two companies tackling the big question of how to make high-quality, patient-specific cells at scale. They're approaching autologous therapy from very different angles, and together we're going to paint a fascinating picture of where the field is headed.
Yeah, we certainly are. So we've got Vladik Krupalnik, who is the CEO of RenewalBio, a company that is focused on addressing the global shortage of transplantable cells and tissues, as well as tackling age-related decline and infertility, really leveraging groundbreaking advances that have enabled the creation of really highly realistic development models from stem cells alone without the need for eggs, sperms, or a uterus, which is incredible. And the company called them Stembroids.
And our other guest is Nabiha Saklayen. She's the CEO of Cellino Bio, a biotechnology company that uses artificial intelligence, laser physics, and stem cell biology to automate the manufacturing process of personalized regenerative medicines. The company's mission is to make personalized cell therapies scalable and accessible to a wider patient population by automating what is typically a very labor-intensive and manual process.
But we always like to ask our guests a little bit of an icebreaker. So your icebreaker question this week is, and I'll ask you, Vladik, first, what's your favorite album of all time?
So I like multiple music types. And like in Russian, we say that a person without rhythm is a person that like an elephant step on his ear. So my mother says that the elephant did a great job and like step on both of my ears. So I like very like different sorts of music, but I think that I practically like one song, which is Starry Starry Night by Don McLean. And I think it's a bit resembling like a journey of founders and scientists.
That's lovely. And Nabiha, what about yourself?
So this is like a good question. So I thought about this quite a bit. I ended up picking an album that I seem to listen to all the time. It is in the French language by Belgian artist Stromae, who is epic. It's like dance, techno, but the topics he's talking about cancer. He's talking about his father leaving him, like very like amazing sort of deep topics. And so I'm going to butcher this pronunciation. Racine Carré, I think, is the name of the album. So check it out. Papa Ute, legendary. I've seen him in concert a few times, and so yeah, I'm saving. I would love for him to be the guest artist when we IPO.
That's great. Well, I guess you're demonstrating more internationalization there, so that's great to be brilliant. Well, I guess I
don't even love French. You know, I grew up speaking German. French is hard for me, but that's how amazing his music is.
Oh, that's great. Wonderful.
Good. I'm going to pick up the album, and I will also cross my fingers that you have that live appearance at the IPO as well. But let's get started by grounding first in the science. So my question first is to Vladik. Can you tell us about the work that was done at the Weizmann Institute and how this led to your Stembroid breakthrough?
Yeah, well, it's an interesting story. It's actually like a scientific textbook. So one puzzle piece leads to another to complete like the full picture of the stem cell regeneration. So Jacob came from the Broad Institute. He did his PhD he did his postdoc with Rudolf Janisch, where he pioneered all the iPS biology, where he showed that every cells eventually come to an iPS, how to cure with iPS technology. And he opened his lab in the Weizmann Institute with the idea to find the missing stage of human stem cells. And this is the naive phase. It's a very early stage of stem cells that have the potential not only to go to all your body tissues, but also for the extra-embryonic tissues. And this was his actually first breakthrough of the lab. And the co-author, or let's say the first author of this paper, is our CSO and my co-founder, Dr. Rod Gaffney, who is running Renewal together with me and also joined the same adventure, having the same phone call, come on, join the ride. And next puzzle piece is the roller culture breakthrough that enabled us to ex utero grow mice natural embryos. And it paves the way for the first time to actually to explore development in a dish, or in our case, in a glass vial that goes spinning around. But again, we could actually see the development occurs in our eyes, with our eyes. And this was the second breakthrough. This is one piece of the puzzle. The third one was that, if you recall, this naive stage has the unique plasticity to form the extra-embryonic tissues. And this is how we made, let's say, the Lego bricks of early development, which became like the final pieces of our venture. And if we integrate all pieces together and we can explore the self-organization of those Lego bricks with the roller culture that enables us to grow them, and this is what actually forms the Regenerative-Technology, or like the most known term for that, stem cell-based developmental models.
So, Nabiha, let me ask you again as a scene setter. I do remember when we spoke, probably maybe 5 years ago. I can remember banks of like drawers, as it were, in the kind of way that you were approaching things. So, as you mentioned earlier, at scale. So where is Cellino's cell culture? And I believe you've got these nebula robots. Where are you with all of that today?
Phil, that's such an awesome reminder, because 5 years ago we had all these beautiful sketches, and I think I was definitely excited, but very scared about whether any of this would work. But it was important to have that visual. I was very fortunate. I did a TED Talk on the main stage in 2021, and it was freaking scary because I was talking about this future of robots doing autonomous level production of stem cells, specifically induced pluripotent stem cells, and it's a super complex process. Many weeks, many months at a time. You start from an adult cell that is very specialized. It's your current biological age. I'm 36, so they're 36-year-old blood cells or skin cells. And then you introduce Yamanaka factors into your adult cells. And it's a very, Vladik, I'm sure you have some amazing thoughts of how to describe it. I think it's like chaos. It's chaotic. It's just like, wow, what just happened? Because the cell is trying to rewind time. So it's doing an epigenetic reset. And that's chaotic. And you get very small number, actually it's a fraction of a percent of cells that are going down this path to get to a pluripotent state. And why is pluripotent exciting? It's not specialized anymore. It becomes your blank canvas where you have maximum optionality. It's childlike because it has so much potential for the future to specialize. And what's been fascinating for me to learn back in the day, also when I was first introduced to iPSCs, is how complex it is to, first of all, drive your adult cells back to a pluripotent state where you've actually gone back in time. You're past your age 0 birth age. You're basically at an embryonic age, which is between day 0 and 14 of, starting, and it's crazy. And then these incredible scientists have to sit and every day watch them, shepherd them, nurture them, decide which cells are good or bad using their magical eyes, hands, and brains. It is a superpower. Literally, they're magicians. And there are very few scientists in the world who can do that. And I've been tracking the cost of iPSC manufacturing for autologous, for personalized. It's come down some, which is great. But back in the day, 2017, it was 1 to 3 million. That's the quote you would get if you called a thermo per line.
Actually, still is.
But we're working on it. Yes. So I have good news there. We've made a ton of progress. So, if you came to Cellino, you'd see like a couple things. One is in the office, a lot of scientists working at our desks. We have a web app that is running the machines doing cell production. So a lot of them are sitting at their desks, drinking coffee, making decisions, looking at how the algorithms are making choices. Everything's good. A lot of our scientists watch the process because it's so fun for them. And they can intervene if they want to, but they don't have to. And that's really cool. You can hit autonomy and it runs for 30 days, 60 days and makes choices. And you test at the end how things went. And we also have these big dashboards that I like to call it my little command center, where you can see how many runs are happening today, how many images were taken, how many algorithmic decisions were made, just a bunch of stats. So we're always tracking what's the larger scale. You went down the hallway into our lab. Yeah, there are these machines generating stem cells. And we're doing a whole bunch of things. And we're going through optimization of like the actual reprogramming, the actual expansion, a lot of stitching together all the pieces. And so far, automation biology has focused a lot on taking off-the-shelf instrumentation and stitching them together. You might have seen these labs. We took some of that. We take some off-the-shelf things, but a lot of times these off-the-shelf machines are not good at recreating human. Well, maybe they're good at recreating human lab operations, but that might not be the best way. So we actually go and we actually build our own machines. So we build optical engines. Those are our high throughput imagers, our laser processors. We build fluidic management systems. But incubators, we're pretty happy with some of the off-the-shelf options. We might build some new ones later. I'm trying to tell my team, let's not build everything. But what's the thing that we build that's special? We build these cassettes, we like to call them. They're these small closed containers that can grow cells in 2D, very highly imageable, laser processable, and it can fit in the palm of your hand. And the idea the vision there is what if we were able to create a world where you didn't need these large clean rooms? If you've ever seen photos of a TSMC fab, giant, right? Huge clean rooms, everybody walking around in bunny suits, and it's expensive to run these large clean rooms. What if you could get it down to be smaller? So we have pushed ourselves, and it's amazing. It's amazing. We were, for the past year, more than a year now, we're well underway on our contract with ARPA-H. And we're thrilled to have the backing of the U.S. government to go for the moonshot. What types of robots and AI do we need to build so that nebulas can make high-quality, personalized, pluripotent cells for all of us in a number of different formats? So we've come very, very far. I'm so proud of my team. And we've hit some incredible milestones at the end of 2025 that, again, were shocking to me and I thought were impossible. That happens to me a lot. A lot of times we set these goals and I look at them and said, humanity has never done this before. Humanity has never run autonomous optical management of iPSCs. Okay, great. We're going to try it. And it's amazing because we did hit autonomy. That was a big moment for us in 2024. And now more to come and more to do. That's one of the other things. It is a journey.
Of course.
The type of scale, I imagine, where millions of humans have access to our own regenerative biology. There's just more to do. You build one thing and you realize there's the next level of infrastructure, which I'm excited by that.
That's great. Well, we can feel your excitement, but I guess we'll need to get into the detail a little bit more of timing and so forth going into market. But Nina?
Yeah. Vladik, Nabiha mentioned, at least for iPSCs, that you're turning back the clock to this day 0 to day 14 time frame. And what you've done on the autologous stem cell side is really hit a very special day 14 breakthrough. So can you tell me a bit about the developmental timeline of your human stemroids and what's going on in the lab?
Sure. So day 14 is like a code name. So this was our first evidence of human developmental models that closely resembled human embryos of day 14. This is mimicking the same compartments, the same structure, same self-organization. And it was all done from iPSCs without any eggs, sperm, or any fertilization. So we took, let's say, a bit before, we took like fibroblast or blood cells. We converted them back to iPSCs to their early origins and then pushed them forward to day 14. So day 14 was the stage that we characterized, and Jacob published the paper in Nature. This is actually was just after the company was initiated. And this stage was the landmark, showing that all distant compartments of development at this stage could be realized, and especially the bilaminar disc. This bilaminar disc, this is actually what distinguishes us from all other mammals. So only apes and humans and a bit of rabbits has this development where we have two discs, that one of them going to be the embryo, one of them going to be the placenta. And this is the stage that it's just before we start to elongate for our spinal shape. And once we moving for this elongation stage, we're passing probably the most crucial part of the development, the gastrulation. The gastrulation part is probably the most important event in our life, even more important than the day we are married or any other day. This is the day that actually those cells committed to be an embryo. So our first milestones is mimicking day 28 stage of human early development. And this is going to be around mid 26, where we currently sort out the last technical issues of the protocol, where we can derive from those stemroids blood stem cells for clinical use, and naturally also other cell types, because everything grows simultaneously in one shot. We will get also muscle stem cells, neuron, mesenchymal stem cells, endothelial cells, and probably many other cells. We just don't know still what we're going to get.
And Vladik, can I just ask you, I mean, the 14-day ceiling sounds like it's obviously a technical limitation, and you mentioned that you're going to be moving to 28 as you hit 2026. Is there also like a sort of regulatory ceiling to that, or are you at will able to do the work that you're doing in this space?
So we're very explicit about the regulatories and ethical boundaries, but with the recent adjustments of the ICCR guidelines, the ones who's writing the knowledge about how to treat stem cells and embryonic or induced stem cells, they are favor of proceeding the work with developmental models, as this is a model. And as you have significant understanding why you're doing that, and it's not for reproduction purposes or any other thing of that sort, but just like to get more cells and to move forward the science, this is approved and in the limits of the ethical boundaries.
Got it. And Nabiha, if I could just maybe ask you from a technical level, how does Cellino combine, the laser editing, the imaging, and AI to obviously take all of this messy stem cell culture and convert that into this high-grade therapy batch? I mean, that again is we're talking about really edgy science from both of your perspectives. So an explainer on that would be really appreciated.
Absolutely. Love that question. So the way I think about it is, when your adult cells are on this remarkable journey to get back to day zero, um, a lot of the changes are very visual. And so you can see that the cells are changing, they're changing size, they have interesting growth rates, they are turning into iPSC colonies. Induced pluripotent stem cells like to grow in a certain way. They like to grow with each other on 2D surfaces. And scientists are so good at looking at these incredible cultures and making decisions about that's a good colony, that's maybe not a good colony. I think this one's better. And it's pretty amazing. So we use imaging, label-free imaging, which is very affordable. It's low cost, and we take a lot of time series data. We're tracking the cells. They are trained on sets of label-free and fluorescently tagged data sets, which is like very popular now, popularized by Allen Institute and Google. We applied specifically to iPSCs. So now we have algorithms that can look at cells and determine in-process quality, which is amazing. So you can do selection, you can yield out bad batches, et cetera. You can choose what to do next. So these algorithms are trained, custom, in-house. And then we use that information to decide, what do you do with this culture next? Should you give it some new media? Maybe it needs more food. Maybe you need to remove some cells here because those cells got too dense. iPSCs love to grow at a certain density. Or these are bad cells. They're actually starting to go down a differentiation path. They're turning into a neuron. Uh-oh, bad. Bye-bye. We don't want you there. So normally a scientist manually would have to passage the cells to change density. So you have to do a surface transfer, which is frustrating because it adds a lot of stress to your whole cell population. So instead of like doing that passaging, we use a laser to generate bubbles. And we kind of hit the bottom of the cell culture surface with a pulse laser, generate bubbles, and you can remove cells and manage density in a super precise way. So we don't do well-to-well passaging, and we like to do laser processing, especially in the early reprogramming days when the iPSC colonies are emerging. You want to like maintain the best ones. Don't disturb them. They're doing their magic. They're going on this journey. And then we have trained algorithms that can look at colonies and tell us, okay, actually the edges are spontaneously differentiating, and then kind of the laser goes in and zaps them away. What's really nice about all of this is that because it's light-based, we don't have to open the container to do any of this. You just shine light. The bottom of the cell culture surface has an absorbing layer that's proprietary to Cellino. Actually, that's sort of my PhD thesis, which is amazing that that's still like core to everything we do. So you get this precision and scale combo that is amazing. So we can image at like mega high volume. We can remove cells at very high volume. So you don't have to do this manual labor, and we can do it now in a closed cassette. So you get just an amazing overall system that, the way it runs in our BL2 lab, going to run the same way in a high-grade clean room, and it should run the same way inside a hospital or halfwhere in the world in Vladik's lab. It just opens up all these possibilities because it's algorithmically controlled. And we love that. Some other things about tech I did want to mention. I was thinking about this over the holidays. So I was thinking about Moore's Law, and it is doubling the transistor on the same surface area. And I think it's exciting because what we're trying to see now is, can we do that with cell manufacturing? And I think we can. But it happens over quite a long time frame. So when I was thinking about it, when I started the company, iPSCs, and in some manual labs, this is still true, it's on the order of $1 to $3 million per autologous iPSC line. It's manual, it's very high cumbersome. And we have our target of getting that down per line to about on the order of $10,000 by 2030. And we're right on that slope. We're right on that slope. Actually, if we went down Moore's Law, it would be more like 15 to 25,000 by 2030, but we have an even more ambitious target. Because what I find is that when we set our eyesight on a more ambitious target, we sort of get closer. So I'm excited about that. Like it's cool. It's one of the things that we actually track with our government contracts, how every technical milestone that we're delivering, how are we doing on that slope? But the level of invention is pretty dramatic getting into the final stages. So every month, every quarter at Cellino is intense right now because we have to think out of the box. We have to, like, we can't assume it's going to just scale in the way we were doing things last quarter. We have to think very creatively. So we're looking at, every couple months now, there's a big sort of relearning that my team is going through. Like, what are our assumptions? What are the bottlenecks? There's a lot of focus right now in QC. There's a lot of focus now on reagent cost, which are becoming the next bottlenecks because human labor we've addressed in quite a significant way with the level of automation. So it gets into the nitty gritty, which is pretty exciting for me. And also what I love about our platform, Nebula, we want it to have the option to be localized and distributed. When you look at semi-fab, TSMC is in Taiwan. There's a lot of geopolitical tensions around Taiwan because of that fab. It is the world's most expensive factory because it's optimized for that footprint with that local workforce. And that's how we get down to the, few nanometer resolution of fab. And I think it's important to build, for us, the inspiration I'm drawing is how do we scale this and how do we make the tech stack to be distributed so it can live in many parts of our country and the world. So yeah, I'm excited. I mean, honestly, all these dots we just connected are pretty wild to me because, when I trained in doing photolithography, it just wasn't obvious to me that that understanding would translate this well, but it's like exciting. So, hey, like in the next couple of years, we really want to be on that, at that Moore's Law cost curve reduction.
I'm definitely rooting for you, Nabiha. The first time I looked on your website and I saw your robot, I really couldn't believe it's a benchtop robot, correct?
Yeah, basically. It's a little taller right now because we have some optimization we're doing. Yeah.
Yeah. And I did just typical average cell culture. So with cancer cells in my PhD, and we had to have big incubators and it was a very manual process. And I'm thinking about how once you make this scalable in the most difficult cells to culture in iPSCs, I really look forward to the day that you translate that into all cell culture and then we're going to have an insane breakthrough in biotech because we're going to be able to bring the cost down significantly across a lot of R&D, which is very, very, very cool. But Vladik, back over to you. I know RenewalBio is exploring developing stem cells into liver, heart, pancreatic, different types of applications. And I know your vision for the company is also to really work on the organ transplantation problem. But I know that's a long journey for now. But where do you see how long that journey is going to take you from where you are today in your preclinical work to your first in human trials?
So I would like to start, first of all, like with the mission of Cellino. So their mission is actually like our first step to go for patients, because if we're not getting our starting material, now in our case, the starting material is the iPSCs, what actually Cellino brings us is that if they want to reduce the costs, so having our procedure makes those treatments less affordable for the patients. So in Renewal, we anticipate to start clinical trials for our blood products. We're going for a rare disease, for a basket of bone marrow failures. One of them is MDS, and the second one is acquired aplastic anemia. We anticipated to start clinical trials around 2028. We're currently conducting the experiments to evaluate the preclinical experiments to evaluate the quality of our cells. We're moving to toxicology and biodistribution studies. And this is our main lead indication currently. But as you said, we're a platform. We can do almost every type of cells. And liver, heart, pancreatic cells. This is our like the not so long-term vision. It's liver cells should be around start clinical trials somewhere around 2030. So we will get them. We'll get there. We're doing it in a stepwise manner because our stem braids needs to grow for the specific stage in which we can derive the proper cells. And it's important to mention that we already have various of type of cells that we're exploring to see how we can use them and transplant them already, even though we are not getting like the real spatial characterized stem embryos. We do have enough cells that have some kind of therapeutical value. Let's say mesenchymal stem cells, we know that there is a problem always generating them from iPSCs for different orthopedical procedures. And this is other businesses that we're trying also to hover under RenewalBio and to look for different partnerships with already companies that using iPSCs. The differentiation protocol may be not the best in class, but we can provide the best in class cells for that procedures.
And Vladik, where does this go in terms of the evolutionary pathway from obviously your first blood products and moving that through to the organ specific cells? I mean, are we talking in the future lab grown organs and maybe even body clones? Is that the potential where you could head in the future?
Interesting question, and I actually get it a lot. So it's critical to separate actually science from science fiction, although in our case, I accept that it's a bit complex to do so. But the platform is not about reproductive cloning. We are not intending to create viable embryos or people. What's realistic is actually using those natural processes of developmental biology to improve the generation of functional human cells and tissues dramatically. We can think about better blood cells, more resistant, better liver cells, better pancreatic cells that can sense insulin in a better manner, and potentially also some kind of organ components, but not whole cloned organisms. Our goal is to solve real medical bottlenecks, not to create humans, but to solve medical problems. We do know that just by supplementing high quality cells, we can address most disease in which the cell function is the root cause of the pathology. I'm also adding that in order to get whole organs, so it's also a feasible target. And in the current environment, we can work with different companies that focusing on the scaffold generation for tissues, where we supplement the progenitor cells as a bioink along with their products to generate and actually provide the real bioink and the biomaterials for bioprinting of organs. This is another vertical that we are looking for. We know that we can supply all of those cells from the same patient in one protocol. And this is one of the fascinating things in our development, that we don't have one protocol for one cell. We actually have one protocol for all the cells. It's like in Lord of the Rings, we have one ring that rules them all. The same is here. We have one iPS that generates it all, actually like what is intended to do. And I think that what's interesting in our platform, and why like people thinking that, oh, we're probably doing clones. No, we're not doing clones. We're not doing reproductive biology. We're generating high quality cells for transplantation. We just use the native and the natural processes of the development of the developmental biology.
Good. Thanks for clearing that up. I mean, somebody had to ask the question that everybody else asked you, so that was my turn today.
Yeah, at least get it once a talk with every person that I get.
I'm sure.
Nabiha, your vision ultimately pushes manufacturing into and very, very close to hospitals. How do you approach safety, standardization, and regulatory compliance when it comes to autonomous systems that are deployed across many sites rather than just one central facility where you'd obviously have a bit more control there?
Great question. The way we've been thinking about it is, well, first of all, we wanted to have an amazing partner. Which hospital system would we want to work with? And we thought a lot about it. We have amazing relationships around the world. And we went with Mass General Brigham. And it's amazing. They're just down the street from us, which is really cool. So there's a lot of capacity to collaborate very deeply and meet in the lab, meet in their clean room, meet in their clinical site, them coming to Cellino, us like running around getting coffee, having lots of regulatory huddles. I do think people ultimately will drive this movement coalescing, and they've been incredible. They're so brilliant. And what we've started with is a Parkinson's program there. It's a phase 1 trial led by Dr. Jeff Schweitzer, who's an incredible neurosurgeon, and had this incredible paper in New England Journal of Medicine, peak of COVID. So I think a lot of us missed it. We were so distracted. But they did the first autologous iPSC-derived dopaminergic neuron transplant in an N of 1 study, which was incredible. But all manual. And what I loved about Dr. Schweitzer's team and GCTI, the Gene and Cell Therapy Institute at Mass General, we're working very closely with both groups, is they understand that this is an infrastructure conversation. They understand that the manufacturing tech has to be scalable. This is an end-to-end autologous product. So starting with patient adult cells, reprogramming to iPSCs, and then differentiating to a final tissue product that is transplanted in the patient inside the hospital. And we came up with this ambitious plan to do everything on site, which I think is dear to my heart because I knew we were going to run into a lot of questions. How to introduce an autonomous Nebula stack into their current clean room infrastructure. Like, how do we operate? Like, how does the regulatory alignment work, et cetera, et cetera. It's been incredible. So tremendous progress. We're moving. So, stay tuned. We'd love to share more on that. One other thing that happened in our journey that was, I would say, my career highlight so far is just building this incredible relationship with the United States FDA. And, we came at it from a manufacturing technology perspective. Of course, we're under CBER, the Center for Biologics, that regulates cell and gene therapies. But we're sort of a manufacturing platform, and most therapeutic developers are running their clinical trials and their INDs. What was fascinating in the past couple years, I would say about 2 years, the regulators paid a ton of attention to talking about infrastructure, and manufacturing was a huge priority for them, which led to them establishing and leading the world in establishing an advanced manufacturing technology designation, which is granted and designated to platform technologies that the US FDA believes are showing high quality production of cells in process control and QC, better decision making, less human intervention, all of the things we just talked about. And we were the second tech stack ever to receive that designation just this past year. So it's amazing. So we have a lot of things moving in parallel, which sometimes sounds like a lot, and it is ambitious. But I firmly believe with my co-founder, Marina, that we have to run all of these in parallel to be successful on the access side. So they're all running in parallel. The AMT designation does create really unique opportunities to engage with the regulators. Any cell therapy that is manufactured on Cellino's platform does have this designation, and it does come with some nice benefits of having deeper dialogues early with the regulators. So we are doing this together. So it is a huge team effort, and I'm just excited to keep building on the manufacturing foundation that we have built and now getting through all those different layers. So Nina, to answer your question, I think we have to be very successful in this first deployment. And it is very ambitious, but we have a killer team. It's amazing. And then we have other therapeutic partners ready to be kind of the next ones to work with. And eventually Cellino will end up having a distributed model. But, I do think iPSC manufacturing will become centralized long term because the facilities management advantages, and then differentiation, scaffolding, doing the personalized sort of ink-based painting of getting the final product out will be done, I think, closer to patients. Depending on go ahead.
So obviously you're through the regulatory discussions, and obviously there's a regulatory road ahead of you. But what are the therapeutic applications that you would see? You mentioned Parkinson's. Where else are you therapeutically focused at the moment?
Yes. So, we, Phil, that's a great question. We focus on being the infrastructure partner to therapeutics developers. We do have some therapeutic concepts that we love, and maybe some point we'll jump in. But for us, we have a very active clinical collaboration on Parkinson's. We have another collaboration with a clinical stage company doing peripheral artery disease, autologous. They're a South Korea, US-based company. We have spinal cord injury that we're working on with Matricel. A couple more that will be shared in the next few weeks. And I would say our strategy has been, because we have very high technical ambition and infrastructure ambition, we do see a huge advantage in partnering with the most advanced clinical stage programs, which right now happens to be Parkinson's. There's a lot of programs in Parkinson's, both on the allo and the autologous side, and it's an exciting time. Actually, I do want to give a shout out. The first conditional marketing approvals are coming up in Japan any day now. Sumitomo for Parkinson's. It's an allogeneic iPSC-derived product. And then there's another heart disease product coming out of a smaller startup. I mean, it's exciting. It's exciting. And I do think the conversation then becomes, okay, allo, here's what's great about it. It's available. It's off the shelf. Drawbacks, immunosuppression, et cetera. What's next? Or what's complementary? What's another option for patients? And then autologous is sort of the next thing. I'm very excited. So yeah, I think we are going to play in a lot of the first wave of products. And actually what's been interesting for me is also to see how our platform can also provide scale, precision, and consistency to even allo batch production. Anything 2D is super compatible with Nebula. So those conversations have also started to come back. It's cyclical. The conversation around autologous, allogeneic, 2D versus 3D, we sort of pushed really hard in one direction for a couple of years, and then we learned some things and look back and learn some other things. I do think all of the options will be available for patients, and it'll come down to like what's acute versus what do I want more long-term, I want to keep taking, what's preventative, what's proactive. So it's all happening right now.
Yeah, that's why talking with both of you is so exciting, because you're tackling the problem from these two different angles. And you both have been able to convince investors also of your vision. And Vladik, you became one of the first biotech investments for crypto-backed YZI Labs. What convinced a Web3 native investor to back your regenerative medicine platform?
So if we look for the philosophical perspective, so long-term thinking, high risk, high impact, building infrastructure for future generation. This is actually what CZ done in the crypto markets. This mindset exists in frontier tech and increasingly in frontier biology. And this is, I think, where both of the parties understood that we have the same aims. Everyone, the CZ and Easy Labs in the Web3, we are in the longevity and in regenerative medicine. And I think it was very natural for him to come and to see us as the spearheads of regenerative medicine, as he did in the crypto. And RenewalBio is a platform company with a big promise to provide various high quality, authentic cells that can change regenerative medicine. And not only that, like, we are not only focusing on longevity medicine, we're also looking for longevity treatments. And actually, most of our investors are more interested in longevity rather than in the regenerative medicine. But we all know that in order to get the drug, we need to go for and to follow the regenerative medicine path. And in fact, Renewal's blood stem cell product is probably the most relevant, and it's probably the proven treatment to halt aging. If we look for parabiosis, when they took elderly mice with young mice, connected them together, and the blood from the young actually halt aging at the older mice. And this is practically the same thing. We provide their own autologous, young, identical, stronger hematopoietic stem cells, blood stem cells. This is what you need to halt your aging. And this is what was so attracting and was a significant interest from various investors, including Easy Labs.
And I guess, Vladik, the amount was undisclosed. Is it still undisclosed?
So we raised about $15 million from NFX, Lionbird, Long Game, Kernel VC, Gangels, and also Easy Labs. And over that, we received another $3 million from the Israel Innovation Authority, which keeps us moving forward to get those liver products to early clinical trials.
Great. Well, thanks for sharing with that. And Nabiha, I guess you mentioned that you've raised about 100 million, right? So with the investor appetite for what you're doing, I mean, do investors understand the money flow? Do they understand the off the shelf process that you're running? I mean, is getting the checks easier, or is it still a challenge out there?
Yeah. So I mean, yes, I did raise a bunch of money. That was exciting and surreal for me, honestly. I think about that sometimes. 2021, I will never forget that year. What's unique about our investor base, Phil, is that they are very aligned on this plan for the future. And I have my two first investors kind of anchor that mentality. So I have Engine Ventures out of Boston. They're on this deep tech mission over how do we change the world over decades by building deep technology. And on the West Coast, the first venture capital I flew out to meet was the wonderful Vinod Khosla of Khosla Ventures. And I mean, I think I said all the things that you shouldn't say in a first VC meeting. Like, hey, I flew out to meet you. I'm so excited. I read about you. And anyway, he was fabulous. He was like, okay, you're a physicist. You trained in semiconductor fab. I'm trying to bring precision to biology. That's interesting. Okay. Let's talk more. Let's get to know each other. He has this unique ability to oftentimes see the future in a way all of us may not have been able to see it, including myself. So my journey working with him is, he always talked about AI since we met. He's like, you've got to introduce AI into what you're doing. I'm like, sure. Okay. Sure. We'll try it. And boom, he was right. And there are moments where he comes to the lab, he comes usually once a year and we show him what's going on and we look at how the system's working. And 2 years ago, he came by, he's like, yes. He's like, this is how cell culture should have been done always. That's great. Like, he sort of knew that, and I feel like I learned it along the way. So that's been fabulous, and I do think we sort of maintain that understanding with my current investor base. And my most recent funding, the $96 million came from venture capital dollars. We've been very capital efficient. We've been focused. Deep tech takes time and patience and all of those things. And then the U.S. government, we have a $25 million contract with ARPA-H. So it's kind of extended our Series A timeframe, but I'm not worried about that. I think it's what we need to do to build something that lasts a century or many centuries. Like that's how we need to think. I'm in Boston. I work right in Kendall Square, but we don't have any traditional Kendall Square biotech VCs. I have great relationships with all of them, but we're just looking for different things and different outcomes. A lot of focus right now in the life sciences market for phase 3 assets, more on the traditional side, not cell and gene. There's a lot of risk. A lot of times the risk is around manufacturing, actually, which is really interesting. And it's been wonderful and hard in cell and gene. You've had these remarkable breakthroughs of cures for CAR-T, for sickle cell anemia, for these incredible gene therapies that are curative. But because they were built on manual manufacturing stacks, we have not seen commercial successes in any of these products so far. I think that's a fair criticism. And my answer to that is, we're building something that we want to scale always. I think I've been fortunate that my investor base has been very patient, because I've said I'm going to go into the clinic when I think it's right, when the tech is ready, because I know it'll scale across many different parameters.
Nabiha, on those parameters, I mean, when you're speaking with investors, does the term longevity come up in any of those conversations, or are people still much more grounded in traditional, albeit new approach, therapeutic models?
It's interesting. My own journey on this, Phil, I didn't consider Cellino a longevity play. We were very much a regenerative medicine manufacturing company, and very much working on diseases that you can run clinical trials on. But there has been a shift. And my shift started, I have a story around this. I was invited to speak in 2024 at Brian Johnson's Don't Die Summit. And I was like, whoa, okay, cool. This is so fascinating. I'd watched his YouTube videos. Okay, and then I learned there at the summit that there's this big movement towards discussing how we solve longevity. And there's the partial reprogramming, the kind of supplement stacking, and peptides is like super hot right now, but also replacement as a strategy. Replacing cells, tissues, and organs is a very important strategy. So that opened up my eyes. And one of the things I can bring to this conversation is something that's very important, actually, in iPSC-based therapies and applications. The age at which you bank your starting material, the cells that become your iPSCs, is kind of important, because as we're aging, we're adding more mutation burden to that starting material. And so the biologically younger you are, the easier, I want to put that in quotation marks, because at Cellino, we've made iPSCs for many, many old individuals and donors. So we can do it, but you still carry that mutation burden. So when I think about long-term infrastructure, having iPSCs generated at birth, or even taking like cord blood that folks are banking at birth and then turning them into iPSCs when the system has scaled, is the way to go. And then having that infrastructure for the rest of our lifetime. So when I reach my 50s, 60s, I know I'll need some pancreatic islets. My whole family has diabetes and heart disease. I'll need some heart cells. We could generate them from my youngest state iPSCs. So they're mutation free and they'll have higher functionality. So I do think replacement is going to play an important role. And like another philosophical thing I've been probing, I'd love your thoughts on this. I do think there's like ton of excitement around partial reprogramming right now.
There is.
And I'm excited about it. I think it's necessary. And what will be interesting to see is how we apply that to organs that are post-mitotic. So they're not really, the cells are not dividing, and we lose that ability to regenerate. So, heart, there are some stem cells in the heart, but brain and heart are two very important organs that we're aging and kind of get locked in and they lose function. The liver is highly regenerative. So that's a good target. So, I mean, maybe the brain and heart will have to be replaced to get us past this 200 age limit. If we get there, I don't know. This is the philosophical discussion I'm like curious to have with a lot of my friends this year, because I think it's going to be a combination of different applications that are personalized to each and every one of us in our own unique biology. So there will be some level of preparation, some proactive interventions, and even like looking at Parkinson's. I do think these first trials are telling us a lot about how the disease can be reversed, or the symptoms of Parkinson's can be reversed with transplant at different stages of disease progression. Like, these are very late stage patients, but like, if you knew you were on that path, would there be a more proactive intervention point, and at what time points do we? So eventually the mismatch, the evolutionary mismatch that we're experiencing, we've already, as humanity, gone past this limit of how long we should kind of be alive. Like, I think like 40s, have kids, raise your kids. Evolution optimized lifespan for reproduction and raising children. But we're way past that, like 70s, 80s, 100s, 200s. I do think this longevity tech stack is going to be, it's incredible. And even with AGI coming online, there's just going to be a lot of overlap of how everybody gets their custom personalized plan for how we live longer and healthier. And like Vladik and I are going to be a part of that stack.
We will be very happy to provide you any type of cells that you need.
Awesome. Sign me up.
You know how to do the iPS, we will do the cells.
That sounds great. Let's do it. I mean, I think it's really about like future vending machines, have them on demand, have them distributed, but all based on my cells. Like, I want them to be my cells because I would want that kind of long-term ability to replace while I'm aging. And so actually, Phil, some investors ask me if they can get like special neurons made that naturally teach them how to do an extreme sport. Like, they're like folks who are
never sleep.
Their level.
Yeah. And I guess, putting the two technologies that you have together, I mean, just extrapolating that a little bit. So Vladik, you would be able to create very early cells that are complete replicas of me. They could go to Nabiha's company, and then in the future they could be banked and then adapted for whatever application I want in the future, which is a beautiful thing. I mean, I guess that's kind of where the joining up comes. Would I be right in that?
Yeah, it's actually that. One company doing the iPSCs, the other company just providing you the whole set of cells that you need. Like, one of our investors, Omri from NFX, always saying that if you treat us as a car, so humanity solved how we can fix cars. So you have an old car, you're going to the shop, you can change the engine, you can change the oil, you're doing a few tweaks, and then you go out of your shop with a bright, better car. This is the aim, like to do the same thing for human health.
And I guess just extracting that one more time, because I mean, you did mention, Vladik, that you mentioned longevity when you were speaking earlier, and that was part of the investor appetite. Nabiha's arrived there from a process of osmosis. You seem to be much more focused into longevity as a theme from what I've seen. Is that right? I mean, are you finding that that's resonating with investors more than perhaps traditional science?
Yeah. So in our case, where our first product is actually blood stem cells, and this is currently the holy grail of longevity. We see how the appetite of investors, and specific those investors that are very focusing on aging, very focusing on longevity treatments, they're coming to us. We had even a series of people that asks, please make our own iPSCs. We want to start the process. We said, okay, let's wait 2 years, at least to start our clinical trials to understand the feasibility of that. But you see that people are actually waiting in line for us to be ready for them. And this is amazing how the whole environment and all the longevity movement even convinced states to provide regulatory benefits. Let's say in Montana, if you just going manufacture your treatment, you can start doing, you can start selling your drug after phase 1. And also in New Hampshire, they have the same system. I heard that in Florida, you just need to have like a very good toxicology report and you can start selling your cells. But you see how the longevity movement of actually changing our thinking of how you make drugs, because they understand that nobody will invest in phase 2 or phase 3 of a longevity treatment. Nobody going to wait for 40 years to understand if it works or not. This is something that the understanding of the functionality is probably going to be on the fly once you're already in the process of that. Yeah, just let's understand that it's safe, but rather from that, let's go and start using it.
Yeah, and just to jump into that, it's a really interesting time. And, Japan has created some really powerful regulatory frameworks where they have their conditional marketing approval that you can get after demonstrating safety in the phase 1. It's very inspired by sort of what's happening in Montana and New Hampshire. So I do think the safety bar is really being met with pluripotent-based therapies globally. Melissa Carpenter wrote a great paper last year about just looking at all the patient safety data from like about, I think 1,200 individuals that were dosed with a pluripotent therapy, and it was very, very strong. I mean, I think that just goes to show everybody was paying attention. We know this is a very powerful cell type because it can become anything, and you don't want any remnants. You want it to be very well controlled. You want the therapeutic grade cell type or progenitor being transplanted to be well controlled. Everything's good. So there's just a lot of moving pieces that I'm very optimistic about. And I'll just say this. I mean, I think one of the things that I love about Vladik's work is the images that you have. I know you've been in a lot of nature and science covers, like more than I can count, but it's very striking, just how the complexity of our biology is coming to life, and what can we do with that from a personalized testing side as well. Not just the transplant therapy, which is amazing, and that's going to happen, but there's just so much to optimize on our personalized biology in a dish.
So this is like one of the things that is actually low-hanging fruits. We also trying to look how we can go and intervene the diagnostics field. Even fetal adverse events. You don't need even the human part, just the mice part. This is actually the first time that we can explore toxicity on fetal adverse effects in scale. If we add like multiple measures of different signals, for the first time, we can actually see and test drug for fetal adverse events in high scale, in thousands of mice stemroids, and not only 20 mice, and then let's go for clinical trials. Now we can actually get the power of numbers.
And all personalized. That's the best part. I mean, I think this is where the conversation changes. When you think about allogeneic versus autologous, we've talked about that quite a bit on this podcast. I think, there's a pharma model that has established itself where like pharma companies are picking targets and they're developing that drug and they're looking at specific cost targets and margins and like what percentage of the population could this drug cover. I think there's a bigger conversation to be had that all of us need to be involved in. And, what do we want for ourselves? What do I want for my future stack of regenerative health that is based on my biology and myself? It is not something that's going to be really discussed in a pharma boardroom. I mean, that's not the type of medicines they've historically built. So I take this community building and infrastructure building and relationship building very seriously. It's one of the things that is like a big philosophical pillar in my life. Like, how do we lean on each other and put together the pieces of the tech stack and the regulatory and the infrastructure and the application? Because that's what it will take to reach the level of personalization that we need for every human being that can benefit from this. So yeah, it's exciting. And I think more conversations, more kind of pushing the boundaries. And I think if we meet again in 5 years, there's going to be a lot of things that have completely changed. Phil, We should do that. Seems like we talk every 5 years.
5 years from now. Well, of course. I mean, and I love the direction of the conversation. So, Vladik and Nabiha, obviously, it's been a really fascinating deep dive. Obviously, how we're seeing these personalized cell therapies are really transforming infrastructure, collaboration, diagnostics, and obviously that leads into extended health span and, of course, longevity. So, thank you so much for sharing your opinions with us today. It's been absolutely fascinating.
Thank you so much for having us. It's been an absolute pleasure. I'm really excited for what's going to happen in 2026.
Yeah,
thank you
all.
so much.
Thank you, Nina and Phil, for having us.
Thank you.
Of course, thank you, Nabiha, for sharing the stage with me and showing.
Oh my gosh, I sent you an email. I want to talk about those stem cells, those blood stem cells. That sounds amazing.
Yeah, sure.
Let's talk.
Let's talk about it.
If our listeners want to learn more about your work, where can they find you? Nabiha, why don't you go first?
I'm on LinkedIn. I share my ideas there. I'm on Twitter at Napsicle. Actually, I'll be launching a Substack shortly, so I'll be on Substack and a couple of other channels.
Awesome. And Vladik, where can we find you?
More about you?
I think LinkedIn is the best option. Most of the time I'm there.
Wonderful. If you enjoyed today's episode, please follow the show and share it with somebody who cares about their long-term health.
So thanks for listening to Longevity Technology Unlocked. Stay curious, stay intentional, and keep investing in your future health. And thanks for joining us.